you for having me. I'm really happy to be here and to give you just a very brief overview of some of the work that's come out of my lab in the last couple of years. We're in my lab. We're really interested in hippocampal prefrontal circuits and working memory, and we use rodent models, as you'll see. Um, so, what is working memory? I think of it as a retention of temporarily useful information. So an example would be the two-factor authentication code that we all know and love. Another example is when we're reading a book, we have to keep track of what's going on in the story. And so working memory is a really important function that we need in our everyday lives. And unfortunately, it's working memory impairments are a common problem shared by multiple neuropsychiatric disorders. And so it's important to study the mechanisms that underlie working memory and to maybe come up with ways of enhancing working memory. And so um, how do we model working memory in rats? We have to use navigation tasks. They have to tell us, so to speak, what they're thinking by going to a place. And so, in these kind of tasks, this is I'm showing on the left a schematic of a task called a delayed alternation task. And a lot of you who have used rats in your research probably have heard of this task. But the rats have to alternate visits to the left and right reward zones in order to get food reward. So it's a memory task because they have to remember where they were on the last trial, which reward zone they visited on the last trial, and alternate to the alternate side of the maze to get a reward. So they have to retain that information about the previous trial and use that memory to guide their behavior on the current trial. And so the first experiment that I'll tell you about was conducted by my former graduate student, now Dr. John Stout. He designed a brain-machine interface in the effort to enhance working memory. And this is one of those experiments that I hoped that it would, excuse me, I hoped that it would work. But I wasn't sure if it was going to. It seemed like a little bit of a crazy out there idea. But the idea was that we would monitor the functional interactions between the hippocampus and the prefrontal cortex using a metric called coherence. And so we had to record simultaneously from prefrontal cortex and hippocampus while rats were on the maze. And we confined them at the base of the maze while we were monitoring the coherence in real time. And the brain-machine interface detected periods of high coherence and periods of low coherence, and on separate trials triggered a trial to occur either on a high coherence condition or a low coherence condition. And so the prediction was that when there was high coherence, or in other words, a very strong functional interaction between hippocampus and prefrontal cortex, that the rats would perform better on the task. And sure enough, that is what we found. So if you look on the right, the bar graph, you can see that the blue bar indicates choice accuracy on the high coherence trials, and the red bar indicates choice accuracy on the low coherence trials. And so you can see that the choice accuracy was significantly higher on the high coherence trials than the low coherence trials. And then the lighter shaded bars are just a couple of our control conditions that I can talk about later if you're interested. Yeah, so the main point here is that that when there was a strong functional interaction between hippocampus and prefrontal cortex, the choice accuracy, in other words, working memory, was stronger. And so that was one way that we came up with to enhance working memory. Another way, and this experiment was done by former graduate student, now Dr. Zach Gemzick. So he used optogenetic stimulation to enhance working memory. So in this experiment, he injected an excitatory opsin in the medial septum. This is a brain area that projects directly to hippocampus and supplies the hippocampus with rhythmic disinhibition, and that basically paces the hippocampal theta rhythm. So it's a really important the medial septal input to hippocampus is a really important for its normal function. And so by optogenetically stimulating the medial septum at that theta frequency, our prediction was that we could enhance working memory in that way. And so we used the same task that I talked about in the previous experiment, the delayed alternation task. The one difference is that we manipulated the delay length. And so we had a short delay of 10 seconds and a long delay of 30 seconds. And then we also had either excitatory stimulation, which was the 6 hertz stimulation. It was the blue laser. And we had control stimulation, which was 6 hertz stimulation, but using a red laser. So the opsin is only sensitive to blue and not sensitive to red. So that was a good control. So as you can see, as predicted, uh, we were able to enhance working memory performance. So let me walk you through the graph on the right. So the 10-second control stimulation, the choice accuracy was above 80%. And then notice that the 30-second control stimulation, the choice accuracy went down to about 70%. And that's normal. So as the delay length is longer, we would expect working memory to be more difficult. It's a bigger challenge to working memory. So we would expect choice accuracy to go down in normal rats in normal conditions. So that happened with our control stimulation. With our excitatory stimulation, though, which was very exciting, what we found is that the 10-second and the 30-second choice accuracy was very similar. So we were able to ameliorate that delay dependent decrease in choice accuracy by stimulating the medial septum and driving medial septal theta or hippocampal theta at 6 hertz. And so we're able to enhance working memory in this way as well. So just in summary, we've been able in the last couple of experiments that came out of my lab, we've been able to enhance working memory through harnessing endogenous prefrontal hippocampal synchronization, and also by optogenetically stimulating the medial septum and driving theta rhythm in hippocampus. And so the implications here are that these findings could lead to the development of therapeutic interventions for working memory deficits in those neuropsychiatric disorders that I mentioned at the beginning. So that's all I have. I hope that was long enough, but I'm happy to take any questions that you guys have about that work.